In this lecture, we are going to cover something called phylogeny, or how do we kind of trace traits between kind of ancestor-descendant relationships. And we do this using primarily genetics. And just so you know, I'm not going to, none of this kind of phylogeny stuff is really included on your exam for this kind of module, but I think that, giving you some background information on this might help with some of the labs that you'll be doing throughout the semester. So phylogenetics is defined as the study of ancestor-descendant relationships. The objective of phylogeneticists is to construct phylogenies, and a phylogeny is a hypothesis of an ancestor-descendant relationship. And what they do is they construct phylogenetic trees, which is a graphical summary of a phylogeny. And they determine this kind of relationship either using genetics or looking at physical traits. So all life forms are related by common ancestry and descent. The construction of phylogenies provides explanations for the diversity that is seen in the natural world, right? So phylogenies can be based on a morphological data, some sort of physical variation. It could be physiological data. It could be molecular data, or it could contain all three. There are instances in which you are using all three of those criteria, and you're seeing if there are any relationships that can be found. Today, phylogenies are usually constructed using DNA sequence data. We use the characters to construct phylogenies. A character is any attribute of an organism that can provide us with insights into history or shared ancestry. In molecular phylogenies, the characters are typically nucleotide positions in a gene sequence, and each position can possess 4 character states. It's either, corresponding to your nitrogen bases, your adenine, your cytosine, your guanine, and your thymine. So this relates to what's called plesiomorphy, which relates to the ancestral character state, the kind of original sequence. You also have apomorphy, which is a character state that is different from the ancestor state, or what we call a derived state or derived characteristic, right? You have synapomorphy, which are derived characteristic state that is shared by 2 or more taxa during due to inheritance from a common ancestor. These character states are phylogenetically informative using the parsimony or Cladisitc criteria. So what we're really doing here is we're creating these trees based on shared derived characteristics, right? What is different that these set of creatures share with each other that is different from the kind of distant ancestor? You also have autapomorphy, right, which is a uniquely derived character trait that is unique to a specific species. You also have a few other definitions that will be important here. You have monophyletic, which is a group that includes all of the descendants of a common ancestor. A monophyletic groups are also known as clades, right? So this is kind of, you can also think about this as like a family tree. It's the entire family tree of a particular species. We have non-monophyletic, which in any case it does not satisfy the above, such as a paraphyletic or a polyphyletic group. And a periphyletic group, you have a group that includes some but not all of the descendants of a common ancestor. You also have polyphyletic, which is assemblages of taxa that have been erroneously grouped on the basis of homoplasious characters, right, i.e. Vultures, right? And sometimes this does happen, but it's not a particularly common occurrence. So look at homology and homoplasy, a character state that is shared between 2 separate DNA sequences or taxa, may be so because they inherited it from a common ancestor, right? Or it is a homologous or homology. Alternatively, the shared characteristic might occur because they were evolved independently, in which case they are called a homoplasy. So if it is a change in the DNA sequence that is the result of something that's shared or inherited from the last common ancestor, then they share it between 2 different species or 2 different set of species, then you refer to it as homologous. If it is an independently evolved characteristic that may be shared with those other species, but there's no genetic link in terms of its inheritance, then you call that a homoplasy. So this is a phylogenetic tree showing the evolutionary relationships as determined by what we call phylogenetic systematics, right? It's how we get kind of in the system A, you have a common land vertebrate ancestor, and it shows you, everyone is kind of genetically related to that common land vertebrate ancestor. But you can even get more complex with it, going to the common land vertebrate, we're looking at the graph on the right here, the common land vertebrate ancestor, coming out, branching out to, amphibians on one side, branching out to mammals on the other. So what this does, it allows you to really kind of start to really parse out when different groups kind of broke off and how highly related different groupings of species are, right? This is how we know that birds today are very highly related genetically to the dinosaurs of the past. And there was even a whole class of dinosaurs that eventually evolved into the birds of today, right? And that we know that those birds are actually very closely related also to certain amphibians as well. So in phylogeny methods, we have phonetics, which is the classification based on similarity in terms of morphology. We have evolutionary systematics, which are ancestors and descendants are organized by the analysis of those homologous characters, right? Whether we're looking at genetic homologous characters or we're looking at physiological homologous characters. And then you have cladistic taxonomy, which groups species according to shared derived characteristics. So looking at evolutionary systematics, this is where time depth is taken into account. And we look at, for this particular chart, we're looking at African ape relationships, in which in this particular instance, based on kind of the end result of the evolution, we would group chimpanzees and gorillas closer together rather than chimpanzees and humans, even though chimpanzees and humans are more genetically related to each other than they are, than chimps are to gorillas, right? Looking at cladistics, on the other hand, which is looking at shared derived characteristics and with no time depth into account, and really looking at kind of more of the genetic information, we group chimpanzees within a sister taxa relationship because they are so genetically related, and we do share a lot of derived characteristics in terms of genetics as well as terms of certain physiological traits that are shared with chimpanzees, in which chimpanzees do not share with gorillas, right? So really depending on which traits you're looking at, depending on whether you're doing a physiological examination or you're doing an examination of genetics, it's really going to alter how you construct these evolutionary relationship trees. And this is called a cladogram, which is a chart showing evolutionary relationships through cladistic analysis, right? It is based solely on interpretation of shared derived characteristics, and there is no time depth taken into account. So how are new species made? Well, we have cladogenesis, which we've talked about a little bit before, which is in which a lineage branches into two or more species over time. Or you have anagenesis, in which there are gradual changes in a single lineage. The ancestor and the most recent are two separate species. So if we look at it in terms of a graphical representation, on the top we have anagenesis, where it starts out as species X, and at some point there are so many changes throughout the evolutionary lineage that it becomes a completely separate lineage by the end, or a completely separate species by the end of it. So the ancestor is completely different than the descendant. Cladogenesis, which is actually much more common, is where you have a single species that ends up branching off into two separate species, right, that become distinct from both the ancestor and each other. So how fast do organisms change? This is called phyletic gradualism, which is essentially saying that changes accumulate gradually over time. And this can be either related to cladogenesis or anagenesis. And what we're really looking at is how does morphology change over time, right? So in essence, in terms of anagenesis, at some point you have that morphology at some arbitrary division, that morphology changes enough to be considered a separate species. And in terms of cladogenesis, usually at some point there's some sort of event that causes two species to become separated from each other and eventually develop into two distinct species in and of themselves. And the rate of change in terms of evolution may vary quite a bit, and this is what we call punctuated equilibrium. It's essentially the kind of tempo of evolution. It's marked by this kind of stasis, right? Nothing's really changing. Then all of a sudden you have a quick burst of morphological change, and then you follow by more stasis for long periods of time, right? So in essence, graphically, it would be represented by, you start out as species X, and then all of a sudden there's a sudden burst of morphological change, and out of that sudden burst comes two separate species, right? So remember that the fossil record is full of gaps. This means that there's really no intermediate morphologies, or we don't really have a whole lot of examples of them. There are different methods of speciation. If we look out in the natural world amongst living species, you have allopatric speciation, which is achieved through complete reproductive isolation, so two groups become reproductively isolated from each other. You have parapatric, which is partial reproductive isolation, so a subsect of the group is no longer reproducing with the group as a whole and begins reproducing elsewhere. And then you have sympatric speciation, in which occurs within a population with no necessary reproductive isolation. In a general sense, we have two distinct, or two kind of really commonly accepted definitions or kind of frameworks by which we define a species. We've already talked about this one a little bit earlier in the semester. We have the biological species concept, which was devised by Ernst Mayr. And he says that a reproductively isolated group of actually or potentially interbreeding populations should constitute a single species, right? But we also have the evolutionary species concept that was proposed by George Gaylord Simpson, which says that a single lineage of ancestral descendant populations should be considered the same species. And what George Gaylord Simpson was really trying to account for was this notion of anagenesis, right? That it's possible for a ancestor species to be a completely different species than the one that we see today, right? This is really showing kind of evolution over time. So something that's very important as we start to move into the semester, where we're going to start exclusively talking about fossils and how fossilization occurs, and looking at different ancient human relatives as well as kind of archaic Homo sapiens, it's very important to understand something fundamental about fossilization. It is a very tricky process, and not all species and not all members of a species will fossilize, right? So only, think about this in two separate ways. First, you have population size versus fossil preservation, right? It's very rare that you have conditions in which a fossil will form. It requires very specific environmental conditions. If you want to review that, go back to looking at our lecture from chapter 8. But so only the most common member of a species is going to fossilize, right? You're not getting the odd, unique variations of that species. You're getting literally the most common one, because statistically, those are the ones that are going to have the highest chance of actually becoming a fossil, right? So only the most common phenotypes within a species might actually fossilize. And then once you go through that fossilization process, you're only getting tiny little fragments of what that species or what that creature actually was. And with that, we'll call an end to this lecture about phylogeny and kind of how we look at genetic and trait distributions in species and how those relationships relate to evolutionary history. So be sure to kind of review these things while you're doing the lab that requires you to kind of construct and build phylogenetic trees. And if you get into any problems, remember I'm always available via email and things like that. Make sure you watch the associated videos with the lab. They'll be very helpful for you. And we'll kind of, from this point on, be moving forward and talking about ancient members of our own lineage, right? The Australopithecines, as well as Ardipithecus ramidus, as well as the genus Homo, right? Early Homo habilis, Homo erectus, as well as a few others, right? So really, as we kind of move through this into this next module here, we're really going to become focused on just our part of the, kind of evolutionary story here on planet Earth.